So hello class, hope you're having a good Easter vacation. Where we left off on in the last video, we were talking about Gregor Mendel and his idea about genetics. So we had talked about how previously people said if you had someone with black hair and someone with blonde hair, that if they had kids, they'd be like a mix. But what Mendel found is that it's not a mix, it's actually sort of chosen by probability which genes, whether from the mom or the dad, is selected. Where we left on is called the rule of multiplication. So if you remember basic probability, if you have the probability of one event and the probability of another event and you multiply it, you get the probability of having both occurring. So for example, the probability of throwing two sixes is the same thing as the probability of throwing one 6 times the probability of throwing another 6. So your chances are 1 in 36. Another thing is called the rule of addition. So rule of addition is a probability that 2 plus mutually exclusive events will occur, and then you add together the individual probabilities. So the ones that we were looking at previously, the rule of multiplication, is what is the probability that something would happen and something else would happen. This one is the probability that something would happen or something else would happen. That's when you add. So for example, what are the chances of throwing a die that will land on a 4 or a 5? Well, because of this keyword or, the chance of getting a 4 is 1 in 6. And the chances of getting a 5 is 1 in 6. So instead of multiplying them, because I'm trying to get an or, you're going to get an answer of 1/3. You have a 1 in 3 shot of getting a 4 or 5 if you're rolling dice. Okay. So for example, here is a things that we've been doing already with the, again, a Punnett square, like the odds of getting the sperm getting the dominant and the recessive gene, or eggs getting the dominant recessive gene, and multiplying this all out, you have a 1 in 4th chance of getting any of these squares. I mean, that's pretty logical. So extending the Mendelian genetics, you need to know that the genotype and the phenotype, the relationship is very complicated. It's complex. Here are some of the things that you need to know, some of these keywords. Complete dominance. Complete dominance is a heterozygote and a homozygote for dominant allele are indistinguishable. So for example, if you have a capital Y, or a capital Y, lowercase y, no matter what, you still have a yellow seed. So this is complete dominance. Like, you don't know, they're indistinguishable which one you have. Okay. Incomplete dominance is the hybrids have an appearance that is between that of two parents. So even though I just said, previously they believed that parents, the offspring of parents, would be the medium between the two, there is a possibility that the mix, so like YY and YY, you get both a yellow seed. Well, incomplete dominance means, for example, in this F2 generation, if I got the recessive of one and the dominant of another, it actually turns out pink. So the ones that we've been studying before was like, hey, you have to get red or white, because the parents are red or white. But they noticed that for some species and some organisms, when you look at their genes, sometimes it is a cross, and they call that incomplete dominance. There is also something called codominance, or a phenotype of both alleles is expressed. So this is also kind of rare. So you could do red hair crossed with white hair, and you have like, I guess here it says roan horses. So you get like both of them expressed. So you have red and white hair. That's called codominance. And then also multiple alleles. So a gene can have more than 2 alleles. Okay. For example, in the example we're going to go over now is like your blood type. So I don't know how many of you have given blood or know your own blood type, but this is actually a very good example of genetics. Okay. So there are 3 alleles for the A, B, O blood types, right? You could be A, B, and O. Okay. And on top of that, there's positive and negative. Okay. So the alleles are IA, IB, and then they call it lowercase I. And IA and IB are codominant, meaning if you have if you have this allele IA and IB, your blood type is AB. Like, you don't get one or the other, you get both. Okay. So here's the blood typing. I'm B positive. You should know that type O is the most rare. So here's the possible genotypes you would have, and then this is the phenotype that you express. So if you have capital IA and IA, or you have a dominant recessive, you'll be type A. If you have a B, B, and a B, you get type B. Anyway, long story short, there are 3 different types, so depending on how you mix them up, you could be a totally different group. And that's part of the reason why when you do blood transfusions, when you donate blood, you can't just give blood to whoever. You can't just give blood to whatever. You have to have the same matching type. So they must match. If you're curious, what happens if you give a the wrong blood type to somebody, the blood actually clumps up and you could die if the wrong blood isn't transfused into your body. Okay. And then there's also something you need to know called the Rh factor. I told you, like, my blood type is B positive. So the Rh factor really is just a positive or negative. So some of these red blood cells, they actually have a protein on them. If it has a protein, that's called plus, or Rh plus, and there's no protein, that's Rh minus. So plus minus, it's still okay, like for some cases, and some cases it's not. But if you need to know, like, what's the difference between A positive and A negative, or B positive and B negative, it's really just is there a protein on the blood cell? A couple more, I guess, examples and definitions. Pleiotropy, a single gene has multiple phenotypic events. So for example, sickle cell anemia. So this is something you should have learned the first time you studied biology. So our normal cells, our normal red blood cells, they look like this. It's really nice. They're soft, they're cushiony, they look like jelly donuts. But in some cases, genetically, people contract what they call sickled cells. And you can see the difference between these ones and these ones is that the sickled cells are very sharp, they're pointy, they're not soft and fluffy. So what could happen is that, you know, the red blood cells can break down. It could actually hurt. So obviously, since they have sharp edges, it can be painful for some people. So this is an example of a single gene that has multiple effects. Okay. Epistasis is when one gene alters the phenotypic expression of another gene. So I'm pretty sure some of you have heard of the term like albino, or things being completely white. So if you have a specific gene, it actually alters the phenotypic events of another gene. So if I display something, it just, it just ruins the rest of the, basically the rest of it doesn't even matter anymore because it destroys it. So for example, if I ever, in this example, I guess these ones are dogs. If you ever get a lowercase e, it overrides any other of the other phenotypes. So do you notice in these down here in the bottom right corner, you get these EEs and EEs. So it doesn't even matter what the first letters, what the B's are, you're going to be albino as long as you have E's. In normal cases, like whatever is dominant, you display it. If you get the black, you get black. If you're brown, you get brown. But if you get lowercase, it doesn't even matter what's in front, you become albino. This is called epistasis. Moving on, trying to get through this really quickly. Again, I'm trying to cut it down under 10 minutes. There's also polygenic inheritance, the effect of two or more genes acting upon a single phenotypic character. So height, skin color, you don't just get one gene and that's like your color. There are literally many genes, many alleles that determine whether or not your skin color is a certain color. That determines what your height is. It's not just one gene. There are multiple ones. In this particular case, there's 1, 2, 3, 4, 5. There's 6 different genes that, depending on what you get, can determine your height or your skin color. And then you need to know that your phenotype is also expressed based upon not only genetics, but also environmental. So you may have heard that, nature versus nurture. So these flowers over here, even though they're red and blue, these blue ones, they actually, depending on what's in the soil, whether it's like acidy or if there's aluminum in the soil, actually could display blue. So not only is it part genetics, but it's also a part what does the environment have. There's an example of a blood typing problem. We'll go over that together. A pedigree, another definition you need to know. We're wrapping up real quick. A pedigree is sort of like a family tree. A family tree that has little icons and stuff that show like what was passed down, what genes, what traits do they have. So all you need to know is that a pedigree, the definition of pedigree, is a family tree that also has designations and icons for traits that are passed down. So lastly, you can do a pedigree analysis. So you could do this with your parents or with your family tree. Things like attached earlobes, or if your hairline is a widow's peak, you could actually chart through your family tree, what is it passed down, what are you most likely to have. And the last thing I want to talk about is there's genetic testing. So now those of you that know people that are having babies, they actually get genetic tested so you could see before the baby is born what possible effects, genetic defects it might have. What they do is they call amniocentesis, so they remove the amniotic fluid and they do the karyotype based on that. And there's also chorionic villus sampling, where they insert a narrow tube in the cervix and they extract the fetal cells to do that as well. So that's all the time I have for you guys today. I'll get to the next video and I'll see y'all later. Take care.